All right, everyone, welcome back. I know people are coming in from the break. You have made it to the afternoon session, to our final and really exciting session of the day. We're going to be talking about therapy development. And our first speaker is Bev Davidson, who has a very storied career. I hope I'm not embarrassing you, Bev, but and is really a luminary in her field. We're really excited to have her with us. She is the co-director of the END Center among her many responsibilities at CHOP, and she is going to talk about gene therapy.
Okay, this is going to be really basic. It's going to go into a little bit more of the background than what you heard from Eric Marsh, and I'm going to go quite a bit slower. So for those of you in the room, thanks for hanging on, and thanks for the kids for hanging on. I know it's tough. So these are my disclosures. And here's the in group that I'm sure you've all seen multiple times over the last several hours. So this is an outline of my talk, and I'm going to give an overview of gene therapy in general, because I think there's some really basic tenets that will help as you all are thinking forward on different either forms of genetic medicine or different therapeutic modalities for your kids. So I'm going to do an introduction to gene therapy, then I'm going to talk specifically about one of the vectors that is used for gene therapy. I'm going to tell you what we know about it. I'm going to tell you what we don't know about it. And then I'm going to share some ways that we in the laboratory and in biotech and pharmacy work to make these things better. So what is the timeline of clinical gene therapy? I thought it was important to put this up for a perspective here. So in 1990 was the very first human gene therapy trial. Now the first gene therapy experiment was done in the mid-60s, where a gene was put into a cell in a dish for the very first time in a heterologous way, meaning that that gene wasn't normally in that cell. And so now we march forward 22 years later in 2012, and that was the first approval. And that's actually quite fast for a new biological therapy, 22 years. Monoclonal antibodies took 30 years. This new gene therapy, Glybera, was so rare and so rarely used that it's no longer even on the market. In 2017, here in Philadelphia, a company called Spark was developing the very first gene therapy in the US that was FDA approved, and this was for an inherited form of blindness. And for those of you that want to YouTube those videos, it is really quite remarkable how the children went from not seeing to seeing. And then in 2025 now, and this year in 2026, we have 8 approved gene therapy products, and they're slowly marching through. But there's a lot of work to be done, because most of the work that is being done to date is using the old style vectors, while those of us toiling at the bench are trying to make newer and better things that are safer. The potential of gene therapy is unlike any other traditional medicine, because it's designed by its nature as a one-time treatment. And it's aimed at targeting the underlying cause of a genetic disease at the very cellular level. It has a potential for a transformative therapy, leading to an improved quality of life. Notice I'm not saying cure. We really worry about the word cure, creating an absolute cure. A treatment that is effective and improves the quality of life is a huge bar to reach, and we're aiming for that. It has a potential for the reduced burden for the patient and for the families, and in costs, because of it is a one and done therapy, and it also improves the adherence to lifetime therapies. You heard earlier about the ASOs. This was a life-changing event. The patient has to go for life. and has a potential for cost savings. If you think about the number of times you visit the clinic cumulatively over a lifetime, it can very much add up. And if you go in once and you ablate the need for those visits, that can be a cost savings. There's very different approaches for genetic medicines, and I think it's important to know what they are. One is gene regulation. Maybe the gene that a patient has inherited there's too much of it or there's too little of it. And there's ways to sort of regulate those levels like a rheostat, like your dimmer in the dining room. And you can use these sort of tools to either silence something that's bad, a toxic gene product, activate something that's missing, or enhance something that's there but there's just not enough, such as STXBP1. There's also gene editing. Dr. Marsh introduced this a little earlier, and that is where you go in and you actually change the DNA makeup of that patient's cells so that it moves from a mutation to a corrected gene. This is very hard to do. And then there's gene replacement, which is what most of us do, particularly for a disease like STXBP1, where we want to put back a missing copy of that gene product. In all cases, no matter what modality we're talking about, the biggest hurdle we have to get from the basic research to the clinical realm and clinical testing to an effective therapy is delivery. For lipid nanoparticles used in gene editing, really the only tissue we can hit well or target well is the liver. For DNA, we want to get to the nucleus for a one-and-done therapy. These adeno-associated viruses that Eric introduced to you are really the way to go. And for antisense oligonucleotides, unfortunately, deep brain targets, which is the case for another disease I work on, those are really, really difficult with ASOs. But some of the more surfacey structures of the brain are a little easier to target. There are different approaches to gene therapy. You can do germline gene therapy, which we do not do here in the United States, but you may have heard of germline gene therapy, which was done in China a few years ago. And that's where that healthy copy of the gene that you inserted into the germ cells is passed along to the children. So we do not do germline gene therapy. It's also known as, for example, eugenics. Hi. We do what's called somatic gene therapy. And here, that healthy copy of the gene is not passed on to the next generation. And gene therapy can be administered inside or outside of the body. Outside of the body is called ex vivo, where you take cells out of the patient, you introduce the genetic material, and you put those cells back in. These are done in settings of bone marrow transplants most often, and are used in disorders such as sickle cell disease or CAR T therapies for cancer, childhood leukemias, for example, where those cells are taken out, they're corrected, and they're put back in. What we generally refer to for STXBP1 gene therapy is in vivo gene therapy, where we're trying to get that gene product exactly where it needs to be, which is the brain or the central nervous system. From vector development to the patient, I've kind of outlined that here for you. First, we have the capsid. I'm going to describe this in a little bit more detail in a minute, so don't worry about reading all those words. And we have the transgene. The capsid is basically the protein shell, and the transgene is what is encoding the STXBP1. Those two together make a vector. We can infuse that material into the blood, or we can inject that material into the target tissue. Some people inject in the muscle, some people inject in the blood, some people inject in the eye, as you heard for that first approved gene therapy. We're really targeting the central nervous system and the brain. That vector then carries that healthy copy of the gene from outside of the cell to the inside of the cell, does what it needs to do to get that healthy gene product expressed. So gene therapy, just to review, what do we know? We know that it could be transformative. We know that you should have long-term follow-up. Most gene therapies are currently designed as a one-time treatment, and the genetic disorder can still be passed on to children. What do we not know? The known unknowns are that the results could be variable. We're not mice. We're all different genetic variants, many different ethnicities, many different reactions to things. The durability of the effect, while very, very long, we don't know how long these therapies will last. So far, we're well past a decade. The long-term safety of these, so far, about 6,000 patients have been treated. There's been less than 20 severe complications. And the full impact of pre-existing antibodies that you heard Eric speak about, all of us have been infected with AAV as children. It's a non-pathogenic virus. We never even know we have it. Usually it comes along when you get an adenovirus infection or a herpes virus infection as a child. And these little viruses they depend on those other viruses to actually make more of themselves. So we've all been exposed now, albeit at very, very, very low doses. But those can make a difference when we're going to go into blood-delivered AAV therapies. And then the immunogenicity of the actual product, because these are delivered currently at exceptionally high doses. And I'm going to explain to you why we are trying to make things that are going to be delivered at much, much lower doses. These immune responses can reduce the ability of that vector to get to where it needs to go, a process called transduction. It can inhibit the gene expression over time. If this is a new protein that's never been made before, your body may say, hey, I don't like that protein. I'm going to make an antibody to it. That's basically the principle behind vaccination. Fortunately for STXBP1, all children have a normal, healthy copy of the gene. They will not make an immune response to that new protein, that extra little bit that we're trying to supplement. It may prevent further AAVs from being delivered, but it's also the case for some brain gene therapies where that's not been the case. And then if your antibodies are really, really high and you're going to go in for blood-based delivery, that could inhibit your ability to enroll in the trial. But again, that's not what we're talking about here. Recombinant adeno-associated viruses, or AAVs, are the most common vector for gene therapy. They lack pathogenicity. Again, all of us have had these infections in a wild-type state. They can establish long-term expression, a one-and-done therapy. They have defective replication. We've completely gutted them. They express no viral proteins whatsoever. And newer tools that we've developed over the last half of a decade and more recently over the last 2 to 3 years have really improved our ability to get to where we need to go in a safe and effective way. These predominantly do not integrate. You heard Eric say that there was a case of integration in a tumor. This is an N of 1, again, over 6,000 treated patients. It can happen. We don't understand why it may have happened. There may be other complications in that patient. And it can be scaled for clinical use and commercialization. Viruses have evolved over millions and millions of years to do what they need to do. And a lot of us would like to think we're clever enough to figure out how to make a new virus in the lifetime of a scientist, which is about 35 years, 40 years. Okay, I'm not quitting yet, you guys. But it's been ages and ages of time that these viruses have figured out how to bind to the right cell and get in and move their genetic material to the nucleus. There's wild-type AAV, and again, we gut that to make recombinant AAV. And that recombinant AAV is expressing our transgene, which in this case is STXBP1. How does it reach the target tissue? I'm going to have a couple slides on this. So we make these recombinant AAV vectors in the lab to carry a healthy copy of the gene. That DNA is then transferred into the nucleus of the cell. It sets up shop there and continues as a little extracellular piece or extra chromosomal piece. It doesn't integrate into your native DNA. And it expresses, again, for the life of that cell. This is why the brain is such a good target for AAV, because there's not a cell division in our brain once our brain is made in utero. And this would not be the case if we wanted to go into the blood. Blood cells turn over every few days. You do not use AAV gene therapy for blood, for example. How does it reach the target tissue? Now, this is really critically important. It binds to a receptor that's on the surface of those cells. And it has to be on those cells at a sufficient level to actually pull that virus out of the liquid that you've infused it into, in our case, the cerebral spinal fluid. That vector will bind there, and then it's internalized into the cell. And once it's internalized, what's really important is it gets out of that little bubble within the cell and goes to the nucleus. This is called endosomal escape and uncoding. And in the nucleus, that capsid is uncoded, that gene is exposed, and it can begin to express the missing gene product that we wanted to. There's different serotypes of AAVs that you may hear about, either in the literature from different colleagues developing things. These are basically like Baskin-Robbins ice cream flavors, right? They're made, for unique ways, and they're really limited by the creativity of the labs that are generating these. More importantly, these vectors can be bioengineered to overcome the body's natural defenses or to reach new receptors that we want them to reach. And we do this by changing the outside, the capsid, and the inside, the DNA or the transgene. To engineer the inside, we essentially use different kinds of ways to express that protein in cells. With STXBP1, we think there are certain kinds of neurons that need to express this protein, and so we want to make sure that this protein is expressed in those kinds of neurons. And in some case, if we're doing editing, we want to optimize that editing machinery so we don't go edit everything around the genome. We only edit the gene we want. And if we need to regulate it, again, we can put these rheostats upstream of that transgene to try and control expression. For the outer coat, and this is something I'm going to show you a couple examples of, we can engineer the outside of the virus to be much, much better at what it does. But if we do that, the key requirements here is that we can still make it. Like we can make it in the lab in a single tube like that, but that doesn't do anybody a lot of good other than treating some cells in a dish. We need to be able to make buckets of it, gallons actually. We need to make sure it hits the right cells and doesn't hit the wrong cells. And that, I think, is something that I've thought about for a long, long time and really devoted a lot of time to. And we need to make sure it's highly potent. I don't want to give you the galaxy's worth of stars to treat a universe. I'd rather give you the universe to treat the universe of cells that is deficient. You heard Eric say that the amount being infused is the number of stars in our galaxy. We don't need that, and that's because half of it doesn't get to where it needs to go, and the other half doesn't bind to the cells we want it to. It's more than half if you think about that great big number he showed. So I'm going to show you a couple examples where we've applied this bioengineering to several clinical problems, where we've engineered the outside of the capsid to improve our targeting. The goal in the laboratory is that we target the right cells and the necessary number of cells to have a therapeutic effect. And keeping in mind how we get there, we start with the patient in mind. How do we get to our target? Our target's the brain. What's the best way to get to the brain? Try and invade the brain with these viruses. Again, not a galaxy's worth, or a universe's worth. And we need to make sure that drugs required for a one-and-done therapy are done in a reasonable and safe way. This wouldn't be the case for a repeated delivery platform. There we would have to keep in mind that access can be repeated, can be repetitively applied. Here for a gene therapy, you go into the right area. And this is our challenge. This is to scale. You see at the very bottom, the brain of a mouse. You can barely see it. Then you can imagine, a dish of cells or an organoid that you may have heard about. A little bit above that is a non-human primate, the NHP, and above that, the human. And then all those colored neurons and other cells within the nervous system are highlighted on either side of that human brain. It is incredibly complex. And again, we need to get to all the pink cells within that area, and we need to not get into all the red cells. And so we have to be very thoughtful about how we design these programs. I'm going to show you here how we developed one program for a circuit-based disease. It's a movement disorder, a mood and movement disorder, where that horseshoe-like thing with a ball in the middle was one target, but so was all the orange areas that are interconnected to that. And we designed a gene therapy that could essentially reach that orange region of the brain. And you notice there's a lot of white all around that. We didn't want to hit that. That was not our target. And our new vector, you can see this blue haze. It basically hits all of that orange area. And the old vector at that same dose, there's a little green dot there. That's how much it hits. And we can quantify that, and you see that we hit anywhere from 90 times better to 530 times better than those vectors that are currently being used in people for gene therapy trials. In another example of a childhood onset disease, we designed a vector to allow for the secretion of a missing protein. And here I'm just showing you, the bars didn't show up. I don't know, for those of you in the front, I don't know if you can see it. They show up really well on my computer. But, they're gray here. They just don't show up here. Okay. So you can see that in that first graph on the left, you see the human child levels, and you see that dotted line across there. With this new vector we way overshot it, way overshot it. This is good news. That means we can come way, 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 way down. And what's outlined in red there is our dose. And then, you notice that the two old vectors that were in clinical trials barely get above that little dotted line or not at all. And those doses are either 10 times more or even greater, and they're still not getting the amount of protein that we need in those brains. So here we have something that's quite a bit better. Well, what about the gene we're talking about today, STXBP1? And I'm showing you some preliminary data from my lab from two scientists in the back, Guillaume Chion and Brandon Brown, who are working in this program and presented the data here earlier during the scientific part of this conversation. And what you can see is that in a wild-type cell with the blue dots, that's the levels of normal. And then you can see even with a really low dose of virus, this brand new virus we made for STXBP1, we have the green dot there. We have essentially equivalent levels to the normal protein. We've already increased it. And so these doses are well under what is normally used to get this kind of gene transfer. And it works in mice, where we're able to correct the levels of STXBP1 in these mice. And when we ask, does it work? You can take our control line and we look at electrical activity. And I want you to just notice that there's a lot of straight lines on here, and it's meaning that the neurons are talking to each other in a synchronous way. And then you look at neurons from our patient here, and you can see that there's a lot more scattering and a lot more lines. And in the patient line that we treat, you can see we've sort of calmed it back down so that it looks more like the normal wild type line. So just to summarize, I hope I've given you a little background primer on gene therapies and the fact that they continue to hold promise. We are making such great tools to begin to go in these disorders. And the vector engineering is really allowing us to accomplish this. These low doses that I'm describing improves the safety. We're orders of magnitude, that means an order of 10 lower than anything that's being delivered currently. That broadens our therapeutic range. What does that mean? That means that if we have to go higher, we can. We're not at the limit where we would tip over the edge and create a toxic problem. It also decreases the cost of goods per patient. I actually oversee a manufacturing facility at the Children's Hospital of Philadelphia. The current therapies cost over $500,000 per patient to make, and we've reduced this down between $15,000 and $20,000 per patient, where it now becomes reasonable to think about moving these therapies forward. So I just want to acknowledge everybody on my team and all of the funding that has helped move this work forward, and thank you.
Thank you, Bev. Does anybody have any questions for Bev?
Hi, I'm Dmitry. If you can go back to the slide with the results of the, yeah, this one. The patient one, it's still, I mean, it looks more vertical, but it's still busy. Does it mean that there is something else going on? The reason I'm asking is that Vera, she doesn't have seizures. She's like seizure-free for a good 10 years or 9 years. But every time we have EEG, they report no seizures but abnormal activities, but they cannot explain what those are. I wonder if these are the...
Correct. This is showing abnormal activity compared to the normal lines that we have. And now the nature of that activity, we're trying to understand quite a bit better. When we put STXBP1 back in this patient's neurons, those abnormal activities normalize. So.
But then what does this, like, it's
what
still
does it,
busy.
It's too busy, and
right.
It's really a qualitative assessment at this point. So what we are also doing are doing EEGs in the mice, and we see electrical changes in the mice. So they don't have a little cap. They actually have 8 indwelling electrodes, because mice will rip off their little caps. And for each of those indwelling electrodes, we can see this abnormal activity, and then we can measure what happens when we put the gene vector back.
Okay, thank you.
Hi, Bev. Thanks for that. It was terrific. You mentioned AAV gene therapy as a one and done. Can you comment on the potential for redosing and routes of administration that might make that possible?
So it's not only possible, it has been done, and those were brain-directed gene therapies for a disorder known as aromatic acid decarboxylase deficiency. And those were essentially using vectors that hit, oops, well, never mind. I guess we started over, so apologies. Those essentially were vectors that hit that deep area of the brain, that brain circuit that I showed you earlier. And those patients were redosed. And it wasn't because the expression waned. It was because the surgeons wanted to better fill the space, and they didn't fill the space with the first surgery. And if you went into the blood, the likelihood of high antibody responses, or into the CSF, the likelihood of high antibody responses are there. But the antibody responses in CSF are in general 1,000-fold lower than they are peripherally. So redosing in the blood is a challenge, although folks are working on that.
Thank you.
Any other question?
I'm going to ask. Hi, Beth.
Hi.
I think a lot of us today are probably feeling the effects of last year, as Charlene started off talking about Capsida and where we were last year with Hope. Going into what you talked about, can you explain a little bit of the difference of what you're doing versus what was happening last year? At one point, you had talked about less than 20% have had any sort of negative reaction from gene therapy, but obviously we have a death in our communities. Can you talk a little bit about that?
Yeah, the unfortunate event that happened last year was a blood-directed gene therapy with AAV, and that the patient was dosed and unfortunately succumbed to the therapy. And I think for that, we understood, it was a fairly high dose again, and for that, we understood very little about what that protein was it was binding to. We knew it was on the vessels that essentially line all of the organs of our body, and that virus had to bind on the outside of the vessels and then get to the inside of those tissues by going across those vessels. And I think there's a lot we don't understand about, there's different kinds of vessels in your body. There's arteries, there's veins, and then there's itty-bitty venules, arterioles, capillaries, et cetera. There's a lot we don't understand about how these big viruses would get across. And so I think the biology, we're still trying to figure out what went wrong. But again, that was a blood-directed AAV gene therapy versus a brain-directed AAV gene therapy where you aren't bothering the vasculature. The other thing I will say is the doses that were used there were pretty high. Not astronomical, but pretty high, much, much lower than we're talking about here today. And I think the other differences were the transgene, how that transgene was expressed, and how that vector product was actually generated.
Hi there, thank you. I just wondered, asked earlier about the antibodies. Mm-hmm. Can you test what antibodies are present? So,
yeah.
Because it also affects, I think, age. I have a 29-year-old with STX, and a lot of times they say, oh, because they're older, they may have many more antibodies than a 5-year-old.
Certainly.
So.
Actually, it might be the other way around.
Okay.
Depending on how many young kids you have hanging around the house. But the antibody levels in the blood are a real issue. The antibodies in the brain, in the CSF, are generally 1,000-fold lower. CSF, if you pull it, it's very clear fluid. Blood is full of proteins, it's full of antibodies, it's full of, many more cells, et cetera, et cetera. But those are easy tests to be done. We do them in the lab all the time. Those aren't clinically relevant, but they do clinical tests all the time prior to a gene therapy. So they can always test. But again, I think it's less of an issue for a brain-directed gene therapy. The bar that if you have a really high antibody titer is higher for a brain gene therapy than for a liver gene therapy. It's like, yeah. Mm-hmm.
Last question. Oh, do you have one? Perfect.
Figure out how to use. Okay, so you talked a lot about the delivery vector and all the research going on there, but the load, the protein, are we done there? Or is that pretty much a known, or is still a lot of research there to figure out?
Yeah, the lab has been working on the right construct to put inside the capsid. We're still testing. I wouldn't say there's a lot of work to do, but we're still testing whether or not we can express it in multiple kinds of neurons versus restricting it to certain kinds of neurons where we think most of the activity is happening. But and then there's other things that we do to the vector that are really important. We reduce its foreignness at in a DNA level, not a protein level, but its foreignness. Like, we don't want the cell to say, "Oh, I got a virus." You know what I mean? So we can play with that a lot.
But you know what needs to be delivered within the vector. You're pretty set on that.
Yep, pretty set on that.
Wonderful. Thank you, Bev.